Question. How do you like to eat a peanut? Before I go further, big apology to all those out there with peanut allergies. I am truly very sorry because I love peanuts, and so do most of the planet as far as I can tell. After millennia of being enjoyed in South America, peanuts got the colonial treatment and were very rapidly spread around the whole world and enjoyed everywhere. And the colonial connections are particularly strong in that it was enslaved Africans who brought the peanut to the United States. Ask Americans today who they associate with peanuts, and you'll probably get either late President Jimmy Carter, who was a peanut farmer before launching his political career, or you'll get George Washington Carver, who does have a very strong claim to peanut fame, but contrary to popular belief, did not invent peanut butter. In fact, Carver was an agronomist, part of the very 20th century American project to use scientific principles to improve agriculture, generally by boosting productivity with automation, fertilization, and irrigation. Carver himself was more focused on techniques that could improve soils, particularly crop rotation, and in supporting black farmers to improve profits by growing crops such as peanuts that can then be processed in a variety of ways to add value. And he pursued his applied research and community education from his position as head of agriculture at Tuskegee University in Alabama for the first few decades of the 20th century. Anyway, back to peanuts and eating them. Maybe, like me, you get a little kick out of taking the peanut, sliding your nails around until you find the place where the two halves meet, and then sliding your nail into the tiny slit as you deftly pull it apart. Inside, tucked between the two halves, is a little nodule. It doesn't look like much, just a little lump of peanut-colored material. If I'm eating peanuts, it's likely they're roasted and salted, which means they're dead. But if you had raw peanuts, you're dealing with something else, not a dead thing, but a living, breathing thing. A seed that is not just full of potential life, but is currently alive. While it's just a seed at this point, it's not doing anything too exciting. It has very low moisture content, and the breathing, or really we should say molecular respiration, is happening at a very low rate. A living peanut is not photosynthesizing, meaning it doesn't have any green leaves to harvest sunlight with, but it is at a very low rate using chemical reactions to break apart sugars, to produce energy, to power a baseline level of cellular processes, including gene expression. And if you were to take that peanut and put it in some damp soil with some overhead light, those dormant biological processes will very quickly ramp up within a few hours. And then within a few days, all that hard molecular work will be easily visible to the naked eye. The changes begin, like I mentioned, long before you can see them, and well within a day. The seed begins to swell, and the cells in the seed coat, that's the thin brown wrapping around the peanut, soften and begin to fall away. Not so much fall away as get ripped apart as something begins to stir from within the peanut. The tiny little nodule, that looks like nothing very much, begins to swell and grow. A tiny protrusion, just a millimeter long in the dry nut, forces its way downward into the soil, expanding to 10 times its original size over the space of a day. This is the radicle, spelled R-A-D-I-C-L-E. The original root, already preformed and hidden inside the embryo at the center of the seed. And at the same time as the radicle is pushing downwards, those two huge halves of the peanut that surround it are undergoing some dramatic changes. They're pushing outwards, separating the soil around the embryo and turning light green. Because you see, what I've been calling the halves of the peanut are the cotyledons, often called seed leaves. And yes, the main body of what we consider a peanut is a set of dormant seed leaves. These are different from standard peanut leaves of a peanut plant, and they play a specific role in germination. That's the process of the seed transforming into a plant. In many plants, the cotyledons push upwards out of the soil and spread out to form fully functional leaves, the first leaves of the emerging plant. But for peanuts, like most plants with large seeds, the cotyledons remain below ground, mostly serving as a store of nutrition for the radicle pushing down into the earth, and for the final crucial part of the embryo, the plumule. The plumule is inside the little nodule between the cotyledons, just where the radicle was, and it's a set of a few tiny true leaves and a stem that push their way up into the light. And at the top of the stem is the engine of plant growth to come, the shoot apical meristem. The seed of a plant is similar to an animal embryo It has a bunch of parts in miniature that grow, but unlike an animal embryo, there is no blueprint for what the final adult version will look like. Animals can develop totally new organs during development, that is true, that are not in the original embryo, but that will all happen according to a specific, predetermined plan encoded in the genes of the embryo cells. A plant doesn't develop according to a predetermined plan in the same way. We say that they have indeterminate development. They have a kit of parts, leaves, roots, branches, flowers, and the exact number, size, and relationship between all these will develop in response to the environment and can continue to change over the whole lifespan of the plants. Well, that does differ a bit from plant to plant, so I'm oversimplifying a little. Some plants do tend to reach a particular end size before reproducing and dying, but still there will be lots of variation in how they get to that end point, the exact number and arrangement of organs they produce along the way. And facilitating that indeterminate, dynamic development are a special pair of organs, the shoot apical meristem above and root apical meristem below. These organs are patches of dynamic dividing cells, and not just dividing, but differentiating as they do so. The meristem always remains, through the life of the plant, always as a center of dividing cells being carried higher and higher or deeper and deeper down as it leaves behind it a trail of differentiated plant organs. We animals don't have this sort of developmental dynamism. We do have some pools of constantly dividing and differentiating cells. In humans, the most obvious example is that for our whole lives, we have pockets of what are called stem cells in our bone marrow, constantly dividing with the daughter cells they produce differentiating into all the different types of blood cells. How can one stem cell in our bone marrow produce all the different types of blood cells? And how can the apical marrow stem cells in a growing plant produce every possible organ of the adult plant? It's possible because every cell in an organism contains the whole genome. So within each cell is the full complement of genes, accessible within the nucleus to an army of proteins that can process information from the outside and use that information to select the most advantageous genes to turn on or off, and in doing so trigger a few of the cells from the meristem to form a new branch here or a new root there, or if the timing is perfect, to produce a flower. Inside the meristem cells, the genes are very busy indeed. The nucleus is thrumming with life. The DNA is furled and unfurled. Proteins called transcription factors are searching along the DNA without hands, without eyes, but guided by the laws of thermodynamics till they find the right DNA sequence. Once they've found the right DNA sequence, they stop moving around and stay in place long enough to recruit the master of ceremonies, RNA polymerase. That's the mega protein complex that runs along the DNA, scanning it and using it to create messenger copies of the sequence made of RNA, the nimble sister of DNA. RNA can leave the nucleus and head off into the cytoplasm of the cell. Waiting in the cytoplasm is another type of megacomplex, the ribosome. Made of a mixture of protein and specialized RNA molecules, the ribosome has a cavity in the side that accepts the messenger RNA molecule freshly arrived from the nucleus. The messenger RNA is fed through the ribosome with the code in its RNA read to produce a protein with a specific amino acid sequence. When we talk about genetic code being read in a cell, this is really what we mean. The RNA polymerase creates an RNA version of the gene, then the ribosome reads the RNA version of that original DNA code. Without a brain, it understands the message, overseeing the assembly of a new protein molecule following the precise instructions that lay within the RNA molecule, which come directly from the original DNA master copy. Once the ribosome has done its thing, the new protein molecule is ready to begin its life. It may live for just a few minutes, or it may live for years. It may be a piece of a fiber cell that will form the internal structure for the cell. It may be one of the hundreds of proteins that facilitate the harvesting of sunlight to produce energy. It may be an aquaporin, a type of protein that sits in the cell membrane, the fatty barrier that separates the inside from the outside of the cell. Given the right cues, it lets water rush in, forcing the cell to expand, a crucial part of all the growth processes we've described. It could itself be a transcription factor, destined to head back into the nucleus to join the dance of DNA furling and unfurling, scanning and reading. None of this could happen outside the context of the environment the seed finds itself in. The water, the sunlight, the minerals in the soil, the bacteria and fungi that surround the seed. In the case of our thought experiment, you, the human, who placed it in the soil, are a crucial environmental factor. These environmental factors are actively sensed and responded to by the cells in the growing plant, with corresponding changes in gene expression. In a sense, the genes are the locus of information processing. With all this complex information about the external environment, the internal condition, and position of the cell all processed into changes to the expression of the several thousand genes in the nucleus. Genes are a physical scaffold within the cell on which information processing can happen. They allow for computation. They are a physical embodiment of billions of years of unconscious generational learning through the painstaking trial and error of evolution. But at their simplest level, genes are pieces of DNA that make the little machines called proteins that make a plant possible. And in this episode, we'll explore just a few examples of specific genes and the proteins they produce, and we'll draw on examples of proteins that are central to plant growth and development. I'll be summarizing the products of research that now spans more than a century, connecting up plant physiology to genetics, and hopefully by the end, it will be clear why genes are an ideal target for anyone who wants to make new crop varieties. Welcome to Modified. I'm your host, Dr. Orlando DeLange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs, and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 4, How Genes Make Plants. If you're tuning in for the first time, I recommend heading back to episode 1 and starting there. Otherwise, let's proceed. Industry for Japan, agriculture for Taiwan. So went an official imperial slogan in the 1920s. This was already decades into the occupation and rule of Taiwan by Japan, after a military conquest at the end of the 19th century. The Japanese imperial government was determined to show how successfully they could operate Taiwan as a colony, and that meant, among other things, investing in agricultural research to turn Taiwan into the breadbasket of the Japanese Empire. So it is perhaps no surprise that one of the greatest discoveries of agricultural science was made in Taiwan by a Japanese scientist in the 1920s. That is the discovery of gibberellins. If you are not an agricultural or plant scientist, I think it's unlikely that you'll have heard of gibberellins, and yet they are at the heart of one of the most dramatic episodes of the 20th century, the Green Revolution. Depending on who you ask, the Green Revolution could be described as one of the greatest humanitarian projects of all time, or a colonial plot to destroy the economies and culture of developing countries. The Green Revolution was a decades-long concerted push to breed new, higher-yield varieties of staple crops, starting with wheat in Mexico in a project led by an American scientist, Norman Borlaug. His work involved applying advances in genetics and plant science, with one of the key methods being to find mutations in genes related to gibberellins. The Green Revolution did boost yields of staple crops. In that process, it also exported American approaches to agricultural production around the world. We'll learn more about the Green Revolution in a later episode, but for now, let's get back to earlier 20th century Taiwan. Researchers using cutting-edge techniques in plant physiology and genetics were hard at work to adapt Japanese rice varieties to the tropical climate of Taiwan, as well as researching the various diseases and ailments encountered that were limiting yields. Among those Japanese researchers studying rice in Taipei was Eiichi Kurosawa. Like many other researchers, he studied the diseases that afflicted rice varieties in this tropical climate of Taiwan. Dr. Kurosawa had taken an interest in a specific disease called foolish seedling disease in English. Rice plants with this disease grew very tall, very quickly, and then would bend over and rot in the mud because they had very weak stems. From studying samples carefully under a microscope, he established that the disease is caused by a fungus, Gibberella fujikuroi. But how? Following an experimental design approach going back to Louis Pasteur, he took some of the fungus, ground it up, and then sterilized it so that he could be sure that none of the fungus cells were alive and active, but that the chemicals they produced would still be present in the extract. Basically, he made sterile fungus soup. No living cells, but all the biochemicals would still be in the soup. And then he applied this fungus soup to rice seedlings. To his satisfaction, they grew very tall, very quickly, and had very weak stems. He showed that there was some chemical that the fungus made that had this effect on the plants, and he named it Gibberellin after the fungus Gibberella. The discovery was not put in a drawer and forgotten. It excited agricultural scientists across Japan, and by the end of the 1930s, the chemical had been purified and its molecular composition studied. And the interest was not limited to the Japanese Empire. Science had been an interconnected global field since the mid-19th century, facilitated by improvements in communication technology. And so, before long, researchers in Europe and the United States were also researching these fascinating compounds. Margaret Radley, a British agricultural scientist working in the UK in the 1950s, was the first to report what had been suspected but not proven till then, that gibberellins are naturally produced by plants themselves. She did this using a similar approach to Eiichi Kurosawa decades previously. She prepared an extract from normal bean plants and applied them to short bean plants from a variety that were consistently much shorter than normal. She found that this allowed the plants from the short variety to grow to a normal height. Essentially, she showed that the dwarf beans were missing some chemical produced in wild-type beans. Studying further, found that this was indeed the gibberellins, the same chemical structure produced by the fungus discovered about 20 years earlier. The fungus was just hijacking an existing system that plants use to regulate their own growth, controlling the production of the amounts of gibberellins produced in plants to promote or reduce growth rates. Specifically, what gibberellins promote is cell expansion. The meristems we met at the beginning of the episode are sites of cell division. However, even though the meristem is full of thousands of cells, the cells in the meristem are tiny. Generally, the whole organ is only visible as a tiny bump on the side of the stem or root. Apply some gibberellin, and those cells will expand dramatically, and the new branch, root, leaf, or flower will begin to grow and form over a period of days. The discovery that gibberellins are produced inside plants themselves wasn't totally surprising in itself. It was already known that another group of chemicals, auxins, could have dramatic effects on plant growth and physiology. In fact, Charles Darwin of evolution fame carried out important early research on auxins. Auxins accumulate in the meristems and are the signal to be a meristem and continue to be a meristem. Remember that the meristem is that cluster of continually dividing cells that can be a site for new tissue and organ formation. With gibberellins added to the mix, it was clear that there are multiple different signals called plant hormones that behave like master regulators of plant growth and physiology. First auxins, then gibberellins, and then as the 20th century wore on, more and more were discovered: cytokinins, abscisic acid, ethylene, brasinosteroids, salicylic acid, and more. They all have a unique set of effects, though there is some overlap between them. Cytokinins encourage cells to stop dividing and to differentiate into a specific organ type. Ethylene causes fruit to ripen, which is a complex set of changes to cell structure and biochemistry that have to be coordinated to all occur at the same time. Salicylic acid triggers a whole suite of changes that allow the plant to better resist pathogenic microbes, things like churning out toxic compounds to kill the microbes. There are about 10 major groups of plant hormone, depending on how you define them. Plant hormones are themselves just one subset of the much larger suite of signaling molecules, chemicals that transmit a signal, a piece of information. All cells and all organisms rely on signaling molecules of some kind. That signal can then be interpreted in the same cell, or in the case of a plant, the signaling molecule can also get moved through the plant in the xylem or phloem systems, or get released as gases from the leaves to be absorbed by nearby leaves on the same or neighboring plants. Signaling molecules are how different cells in the same plant talk to each other, and how neighboring plants or even organisms from different species can talk to each other. Plant hormones are small organic molecules. The atoms in a plant hormone do still form quite complex structures with oxygen and hydrogen atoms, as well as some other elements like nitrogen and of course carbon, but they are small and they are made of 10 to 100 carbon atoms linked into a molecule, as opposed to the tens of thousands of carbon atoms that might be in a single protein. Proteins, like DNA, remember, are polymers. And with polymers as our hook, let's recap quickly on DNA, proteins, and genes. It's pretty important stuff. It's worth going back over. DNA is a polymer made of units called bases. Proteins are polymers made of units called amino acids. There are 4 different types of bases that are all similar to each other, with subtle but important differences. And without those differences, you wouldn't be able to have information encoded in DNA. through the precise order of the bases A, G, C, and T along the DNA polymer. The 20 different amino acid types that make up proteins have a common core unit and then dramatically different side chains, meaning the variable section of the amino acid that literally sticks out of the side of the chain of amino acids. In proteins, these amino acid differences are crucial. They are the magic ingredient that lets proteins do what they do. The different side chains of amino acids have different physical structures, allowing them to interact in different ways with other molecules, and they have different chemical and electrostatic properties, allowing them to facilitate different kinds of chemical reactions. The different properties of each amino acid type not only change how that specific amino acid within the larger protein can interact with other molecules, but they crucially affect how all the amino acids within a single protein chain interact with each other. Some amino acids attract each other, some repel each other. That mixture of attraction and repulsion causes the chain to fold up into complex 3D shapes. And then very often, multiple amino acid chains will actually come together to form a stable megastructure. An individual amino acid chain in a living cell could have anywhere from 10 amino acids to tens of thousands of amino acids. Still, that pales in comparison to the hundreds of millions of DNA bases in a typical chromosome, which, if you remember, those are the things that we have a bunch of in each of our cells, and each one is a single massive DNA chain. A typical protein is a chain of a few hundred amino acids. So how does that relate to the hundreds of millions of DNA bases in a chromosome? What's the connection between them? Each chromosome is a giant unbroken DNA chain, like I mentioned, but it contains thousands of separate overlapping units that are what we call genes. And each of these genes is much shorter. A gene has a few functional sections to it, one region at the physical beginning of the gene called the promoter, that recruits the molecular machinery necessary to make the RNA copy that then gets fed into a ribosome to churn out a protein. Then the middle of the gene is the coding sequence. This is the bit that encodes the protein. This works via a 3-to-1 code, 3 DNA bases per amino acid, which is necessary because there are only 4 different DNA bases, but 20 amino acids. And then there is the terminator, which has signals that stop the RNA production process. Basically, the gene is a set of DNA bases that encode a single protein, plus other DNA either side that has important signals read by that machinery that turns DNA into RNA. That machinery is actually a set of dedicated proteins. The process of turning DNA into RNA is called transcription, and so the proteins involved are called transcription factors. So let's connect this back to gibberellins. Those are small organic molecules which are not proteins. So how do they get made? Are they also encoded by genes? Yes and no. By the 1930s, when gibberellins were being categorized, it was already well established that a lot of biochemistry inside cells is facilitated by things called enzymes, and that enzymes are proteins, made of the same basic amino acid building blocks as structural proteins in fingernails and muscle fibers. Unlike those structural proteins, the proteins that we call enzymes have complex folded shapes that often have a physical pocket that has just the right space and configuration for one or multiple molecules to make contact, and then within the pocket, one or a few specific amino acids will facilitate a chemical reaction that either makes a new chemical bond or breaks one. The details change a lot, but that's the basics of how an enzyme works. And without them, there would be no life on Earth. Because life on Earth relies essentially on chemical reactions that would never happen if you just let all the molecules involved float around interacting randomly. Enzymes allow structure and direction to exist within networks of chemical reactions, rather than chaos and a constant back and forth of making and unmaking. So even though cells are full of diversity of molecules, not just DNA and proteins, they all indirectly connect back. Proteins, specifically the subset we call enzymes, direct the biochemical reactions of the cell, and those proteins are all manufactured in the cell according to the blueprints encoded in DNA in functional subunits we call genes. So where did we get to so far? We know that there are things called gibberellins. They are small molecules that are made by plants, and they signal to cells that they should grow and expand. Like all small molecules in the cell, their synthesis must be controlled by enzymes. All of that was clear by the late 1950s. Decades of plant genetics research had yielded plenty of mutant varieties that were shorter than normal, which made it simple to start testing whether the mutated genes had something to do with gibberellins. That was exactly what Bernard Phinney did. He was an American plant scientist working at UCLA in the 1950s. He started taking mutants, for example, dwarf mutants of maize. That's the name for corn used by plant scientists, because in other English-speaking countries the word corn is used for wheat. Anyway, he found that in many of those dwarf maize mutants, purified gibberellins would cause them to grow to normal height, similar to what Margaret Radley first demonstrated a few years earlier. But this time with purified gibberellins, so he could be sure that it was those exact molecules that were responsible for the effect. And by carefully studying the behavior of the different mutants in response to carefully externally applied gibberellin, Phinney was able to start piecing together a story about how gibberellins work in plants. Over the coming decades, his lab and others showed that there were a whole range of genes involved, each one encoding a different protein. Some of these genes encode the enzymes that actually synthesize gibberellins. If those are broken or mutated, the plant will not be able to make its own gibberellins and will be short, but it will respond perfectly well if the gibberellins are applied externally. Other genes encode the proteins that detect gibberellin in the cell. Similar to enzymes, proteins like this, called signaling proteins, often have pockets to capture small molecules, and then binding of the small molecule will trigger some change in the protein. For instance, shifting its structure to allow the protein to move from the main area of the cell, the cytoplasm, into the nucleus where the DNA is. Inside the nucleus, there'll be a waiting transcription factor. Remember them? The transcription factor will bind to the signaling protein, and together that will trigger some change in the transcription factor and its function that will allow it to turn some target genes on or off, up or down, aka the hormone will trigger changes in gene expression. And if any of those genes, the genes that encode the signaling proteins or the transcription factor, if they are broken, then the plant will not be able to respond, at least fully, to externally applied gibberellin, no matter what. And then there are the genes that are turned on or off, up or down by the transcription factor, the target genes themselves that produce the observable response in the plant's growth and physiology. For gibberellins, a lot of these encode cell wall modifying enzymes. You are probably aware that plant cells, unlike our own cells, have rigid walls, which give them shape and physical strength. These cell walls can be relatively thin, or they can be very thick and embedded with strengthening molecules like lignin, which is the main constituent of wood. Plant cell walls can be very diverse from cell to cell and plant to plant, and crucially, they are dynamic. They can be made and unmade, strengthened and weakened, and if a cell is to expand to get bigger, then they can be loosened to allow this, and then made more rigid again at some point, like loosening a belt and then refastening it. The main structural components of the cell wall are not proteins, they are sugar-based polymers, cellulose, as well as the aforementioned lignin. But it is proteins that control the production of these polymers. Proteins help to ferry all the building materials from inside the cell to the outside where the wall is. The wall sits just on the outside of the cell with lots of cross-links keeping it attached to the cell. Gibberellin master response regulator proteins, those transcription factors that get activated by gibberellins, when they are activated, they turn on genes that encode cell wall remodeling enzymes that can loosen the belt of the cell wall, allowing the plant cell to grow. That is not the only thing that gibberellins do, but is one important part. And so you can imagine if those target genes, such as the cell wall remodifying enzymes, are broken, then you'll see that some part of the gibberellin response is completely lacking. You might find that when you apply gibberellin, there is some element of the normal response, but not the whole response. Hopefully, if you weren't already, you are by now convinced that no matter how inert plants may look at times, they are a dynamic hive of biochemical activity, with genes and proteins driving it all forward, and that plant scientists have been picking this all apart in detail for about a century, and in the process, finding ways to turn the molecular levers and hijack the biochemical pathways to meet human ends. Developing crop plants that will, depending on your perspective, feed hungry people and create a tasty bounty of produce, or subvert traditional food systems to further the interests of imperialists and capitalists. Well done for making it through what has been an episode dense with information. In our next episode, we'll pick up by looking at some of the genes involved in defending plants against microbes, recapping and reinforcing some of the key concepts and facts that we've covered in this episode. Since this one has been so dense with facts, I think a little recap of take-home messages would be helpful for everyone. Number 1. Plant development is indeterminate. It doesn't follow a predetermined master plan that says how many roots, leaves, or flowers the adult plant will have. Instead, meristems are sites of new organ formation that are present within the embryo and are retained through the life of the plant, with additional meristems forming as the plant grows to allow for side branches and side roots. Number 2. Information about the environment and about the states of the cell are processed and transformed into changes in gene expression. This is common to all cells, not just plant cells. Number 3. Plant hormones are master regulators of growth and physiology. Examples include auxins and gibberellins. Number 4. Proteins are chains of amino acids. Amino acid diversity allows for proteins to have a huge possible range of structures and functions. Number 5. We can group proteins based on their functions. For instance, enzymes facilitate biochemical reactions, including the synthesis of plant hormones. Other proteins regulate gene expression, and we call them transcription factors. Thank you for listening to Modified. This was episode 4, How Genes Make Plants. It was written by me, Orlando DeLange. I used many sources to help me research this episode. If you're interested in learning more about the history of Japanese colonial agricultural research in Taiwan in the context of global colonial history, then check out the 2020 article Horai Rice in the Making of Japanese Colonial Taiwan by Wei-Li Leow, linked in the show notes. This is a tiny one-person podcast, so please, if you're enjoying it, subscribe, leave a review, and tell a friend. And if you have notes, well, I'd love to hear them, and you can email me, orlando@modifiedpod.com, and I'd love to hear from you. I'm actually planning to add an extra episode in a couple of weeks, just devoted to answering listener questions. From episode 6, we'll shift gears to talking more specifically about crop plants and agricultural science. But before then, between episodes 5 and 6, I'll be doing a special episode answering your questions. So send me in your questions about genetics and plant science generally, and I'll pick a few to dive into in detail in a special episode. So that's your questions on genetics or plant science, email to orlando@modifiedpod.com. And as always, you can find out more about me and the podcast at the website modifiedpod.com. That's MODIFIEDPOD, all one word, .com. Thanks for listening. Until next time, bye!